Hi, this is the second video on cell physiology. On this video, we're going to focus a little bit on the organelles of the cell and discuss their function. We're going to start with the ribosomes. Assuming I can get my pen working. So ribosomes are made up specifically of a type of ribonucleic acid called R ribonucleic acid, R standing for ribosome, so ribosomal RNA and proteins. And we'll talk more about RNA and DNA in the next section. So for now, just know that they're made of RNA and proteins. And the main function of ribosomes is to actually make proteins. They're located, the ribosomes themselves are located within the cytoplasm, and they can either be found free-floating, so in other words, floating in the cytoplasm and not attached to anything, or attached to membranes, specifically the endoplasmic reticulum. So if they're attached to that, it is referred to as the rough endoplasmic reticulum. Because it's rough, because it's got bumps, it's had these little ribosomes attached, also referred to as RER, the rough endoplasmic reticulum. The ribosomes themselves are made by a structure called the nucleoli, which is in the nucleus. So, for example, if this is a drawing of a cell, and here's the nucleus, you'll often see these little darker areas within a nucleus, that's a nucleoli, that makes the ribosome. And then the ribosomes themselves are located out in the cytoplasm, either free-floating or on a membrane structure called the rough endoplasmic reticulum. And the main thing to remember about them is what they do is they produce proteins. So there's two types of endoplasmic reticulum. The rough endoplasmic reticulum, which this is a picture of it here with the ribosomes attached, and what's called the smooth endoplasmic reticulum. Darn it. Darn it again. Let's try that again. The smooth endoplasmic reticulum, which is smooth because it doesn't have ribosomes attached. And the function of the smooth endoplasmic reticulum is it makes some cholesterol. So one of the functions is to make cholesterol. Another function is it helps to break down fats in a cell. But the one that's probably better known for is it does what is referred to as detoxify drugs. So for example, let's say you were to take Tylenol. You have to remove Tylenol from the body in some way, and the way it happens, sort of in a nutshell, is the liver has enzymes that convert the Tylenol into a substance that you can easily remove from your body by urinating. And these enzymes are often enclosed within the smooth endoplasmic reticulum, and they are detoxifying that Tylenol so you can get rid of it. So organs such as the liver is going to have a lot of smooth endoplasmic reticulum because its function is to help you detoxify or remove drugs from the body. Other organs that don't have that function have much less smooth endoplasmic reticulum. Another organelle that you might be familiar with, heard of before, is the mitochondria. Oops. And the mitochondria is another double membrane structure. So if you have your cell and your nucleus, You might have a mitochondria out here. They're in the cytoplasm. They also have two membranes. So they have what's referred to as an inner membrane and an outer membrane. And inside of this mitochondria are enzymes whose job it is to take glucose, once you get it into the cell, remember with the help of that insulin, and convert that glucose into ATP. We will talk a little bit more about how this process actually happens next semester, primarily actually, but the purpose is to take glucose and convert it into ATP, and ATP is the energy unit of the cell. It's the unit by which we do all cell activities. The process of converting glucose into ATP is called oxidative phosphorylation. Oxidative because it requires oxygen. So to convert glucose to ATP, you must have oxygen in order to do that. Phosphorylation because basically what you're doing is you're taking ADP, which D stands for di, so I think of it like this. It's adenosine diphosphate. It has two phosphates. You're adding another phosphate to that to make ATP, which is adenosine triphosphate. So it's got three phosphates. So you're using oxygen to add a phosphate to ADP and make it into ATP. And you do that with glucose. So glucose is where the phosphates and everything come from. So that's conversion there. Not really liking the way that sounded. I want to restate that a little bit. So what I wanted to restate is that glucose is broken down. You're converting it using the energy from glucose to produce ATP. So glucose itself is made of carbon, hydrogen, and oxygen. There aren't any phosphates in glucose. But as you break down that glucose, you release energy. And you use that energy plus oxygen to convert ADP into ATP. So you're using the energy from the glucose and oxygen to add a phosphate to ADP. Hence, it's called oxidative phosphorylation. That sounds better. The next organelle we're going to talk about is the Golgi apparatus, which I think of as the post office for the cell. And basically what the Golgi apparatus does is proteins enter the Golgi apparatus. So here's your Golgi apparatus. Proteins come in, and then they exit the cell in packages. So they either exit the cell in what is referred to as a lysosome, which is proteins contained within a membrane that are going to remain in the cell. So this particular group of proteins will be in a membrane and they will stay in the cell. Or they're packaged in what's called a secretory vesicle, which again is also a membrane that surrounds proteins. But in this case, these are proteins that are going to exit the cell to be used by some other cell. So proteins come into the Golgi apparatus and then they're packaged for distribution. Some of them into what are called lysosomes, which remain in the cell. some of them into secretory vesicles which exit the cell. So a little bit more about lysosomes. So the function of lysosomes, again, remember they're proteins, and it's proteins that are surrounded by a membrane, and they often are these proteins are enzymes. So remember, an enzyme is a type of protein. And in this specific case, they are enzymes that are referred to as proteolytic. So lytic means to break. Proteo refers to proteins. So the function of the enzymes in the lysosome is to degrade other proteins. So I'm going to give you a concrete example here that might help. So we talked about at the beginning of this section that white blood cells will sometimes have extensions of their cell membrane that are capable of bringing bacteria into the cell for the purposes of breaking down that bacteria. So you got your white blood cell, you have your cell membrane extension, you have your bacteria. So once that bacteria is brought into the cell, here it is, it's inside a little vesicle. And here is a lysosome, which I'm going to abbreviate with an L. So that's a lysosome. So what happens is these two organelles combine. So you end up with a membrane that has the lysosome in it and this nasty bacteria in it. And these lysosome enzymes break down that bacteria, the proteins. So they're proteolytic. They break down the proteins in that bacteria and destroy it. So this is one way that white blood cells are capable of destroying pathogens. Another interesting organelle is what is referred to as peroxisomes. And peroxisomes are capable of destroying free radicals. So we talked a little bit about free radicals in the last chapter. And free radicals are, if you are not remembering, they are compounds that typically have oxygen, and that oxygen has an unpaired electron. So electrons are supposed to be paired. If they're paired, they're happy. If they're unpaired, then they're unhappy and in search of another electron. And they get that other electron by stealing them from other molecules. They might steal the electron from DNA, which can cause mutations. They might steal the electron from fats or proteins in a membrane, which can destroy a membrane. So free radicals can be very dangerous. So what peroxisomes do is our cells all have peroxisomes, and the job of peroxisomes is to convert these free radicals into harmless substances. So they're able to collect up these free radicals and make them so that they will not harm the rest of the cell. Excuse me. Another type of organelle we're not going to spend a lot of time talking about right now are referred to as centrioles, and their job is to help with cell replication. So we'll talk about that a little bit more in the next section. So the last bit I want to review here, talk about, is all the organelles that are involved in protein function and transport. So just to refresh your memory about this, proteins themselves are made on the ribosome. So that's what makes the proteins, and we'll talk about how that process works in the next section. Then they are transported via the rough endoplasmic reticulum in what is called a transport vesicle. Vesicle just means membrane-bound substance to the Golgi apparatus. Once they get to the Golgi apparatus, then they are repackaged into what are referred to as lysosomes or secretory vesicles. And a reminder, the lysosomes stay in the cell, and their job is they contain enzymes, specifically proteolytic enzymes. The secretory vesicles, on the other hand, their function is to exit the cell, and that protein is used by some other cell. So if you look here, here's a secretory vesicle. It binds with the plasma membrane and then expels the contents, the proteins themselves, into the extracellular fluid. So for this video, you should know the function of each of these organelles, and in regards to protein synthesis, know the function of the organelles involved in that and the order in which proteins are moved or shuttled through or by those organelles. So ribosomes to rough ER in a transport vesicle to the Golgi, and from there to lysosomes or secretory vesicles.